Hello everyone, here's the beta version of the first lecture. I don't know how it will be, but let's try. Well, the first lecture will be about the culture media and about how do we grow the bacteria, how do we cultivate the bacteria in the microbiology lab. First of all, what do we need to know about the bacteria is which kind of bacteria we need to grow, when do we need them to grow, which is the purpose of cultivation, of isolation of different pure cultures, and so on. Well, in medicine, usually we do it for diagnostic purpose. That means whenever we need to find out which kind of infection we have, whenever we're talking about bacterial infection, we can have two different ways of cultivation. First of them is a direct one, so it will be bacteriological cultivation, and the second one will be indirect, and it is regarding immunological tests. So now we'll talk about direct method of diagnosis in bacteriology, namely bacteriological method of diagnosis. Well, in bacteriological methods, we are using different culture media. The culture media, as well as you could see, is a kind of nutrient for bacteria which can help it to grow. Of course, the nutrients, it is not main requirement to make possible cultivation of bacteria. As well as you know, especially pathogenic and conditionally pathogenic flora needs specific temperature. And this specific temperature is the temperature of the human body, 36-37 degrees Celsius. Well, whenever we will cultivate the bacteria in the lab, it will be one of the main requirements to create proper temperature for these bacteria. For such purposes, we have special devices. Special device we call thermostat. It's a special box in which we will leave Petri plates or tubes with culture media, and this device will keep the same temperature during the required period of time. And this period of time will depend on type of bacteria we are using. For example, if we need to cultivate some simple bacteria, some conditionally pathogenic flora, it will be enough to leave them growing by night or about 1 or 2 days in order to get the required result. Meanwhile, we have the bacteria which are growing slowly. So, for example, Mycobacterium tuberculosis, which is acid-fast bacteria, this kind of bacteria will grow slowly. So it could take about 30 or even 40 days to grow such bacteria. in the culture media. So for different bacteria we have different type, we have different time required for cultivation. So we will put the Petri plate, or we'll put the tube with culture media in the thermostat for that required period of time. Meanwhile, the bacteria will grow. So, which is the composition of culture media? You can see it here. It's a water, it's an energy source, carbon source, nitrogen, mineral salts, and special growth factors. Not all of them and not every time will be present in every culture media, but usually the majority of them will be present there. As well as life is impossible without water and without energy. And of course, without proteins. Well, it is the main data about culture media. So if we will talk about classification of culture media, we can have classification based on physical state. It will be solid media, semi-solid media, and liquid media. Solid and semi-solid media is composed of solid part and nutrients. But liquid media is composed of water and nutrients. Well, the solid part of the solid and semi-solid media is composed of the substance which is called agar, and it is the substance extracted from some sea plants. Meanwhile, liquid media, as well as I told, is composed of water and nutrients. Also, regarding the ingredients of the culture media, we can have simple media, complex media, synthetic media, and special media. Well, as well as I told, the solid media is composed on such substance which is called agar. So whenever we are talking about semi-solid media, it is mostly useful for demonstration of motility of the bacteria, and namely here you can see two different tubes with solid and semi-solid, with two different tubes with semi-solid media. Both of them were poked with bacteriological loop. So in this case, these bacteriological loop were containing non-motile bacteria. So the bacteria started growing and multiplying just on the way of the entrance of this loop. Meanwhile, in this tube, in the right tube, you can see that the place where culture media were poked by the bacteriological tube is not clear enough, because bacteria were able to move, not just growing on the way of the loop, on the way of penetration of this loop, but also it started growing inside the culture media because it is semi-solid. The bacteria is able to move a little bit more, not just on the way. Whenever we are talking about liquid media, you can see almost transparent but a little bit yellowish liquid in the tube. So how can we see the growth of bacteria in liquid media? It will depend on type of bacteria. Some of them will make all the entire media will be full of bacteria, so these bacteria will make the liquid to not be transparent anymore. In other cases, bacteria will just floating on the surface of the culture media, so it will be kind of like film on the surface. And sometimes the bacteria will be kind united inside the culture media, so it will be like a cotton ball inside this culture media. So whenever we are talking about simple media, we are talking about media which just helps us to grow the bacteria on the Petri plate. It doesn't have any specific nutrients except the main nutrients extracted from meat and the base which makes the media to be solid, which is called agar. Whenever we are talking about complex media, we are talking about media which helps us to make something more than just growing and cultivating of bacteria. For example, this example of culture media, which is called blood agar, which has extremely, which has a lot of ingredients, and it will show how bacteria could act inside the body. Whenever we are talking about synthetic culture media, it is a culture media which is produced just by using chemical substances. So we just synthesize the required components in the culture media. By talking about special media, they have different purposes. For example, first two media, enrichment media and selective media, they have quite same purpose, but they have opposite mechanism of action. Whenever we are talking about enrichment media, we are talking about media which is provided by which is surplused with specific nutrients which will help to grow one kind of bacteria. Whenever we are talking about selective media, it also contains specific nutrients which will help to grow one kind of bacteria. So in the beginning you could have a question, which is the difference between enrichment media and selective media. And here's the answer. The answer is in the question. The answer is in the name of the culture media. Whenever we are talking about enrichment media, we use it just in cases we don't have enough amount of bacteria in order to study it and to see it. So in cases in which we are making diagnosis or we are cultivating some kind of bacteria and we don't have enough amount of bacteria in order to do all the required works and all the required investigations with these bacteria. In such cases we are using enrichment media. So in this case we have just one kind of bacteria which we need to grow, to multiply, and in such cases we will apply specific enrichment media which will help to grow one single kind of bacteria. Whenever we are talking about selective media, we are talking about cases in which we have too much different bacteria. So in both cases, enrichment media and selective media, the media will help some specific kind of bacteria to grow. But in first case, if we have just one kind of bacteria and its amount is not enough, but in second case, we have a lot of different kinds of bacteria, but we need to select among them just one. So enrichment media, when it is not enough and we need to enrich it with bacteria to multiply them, but selective culture media is whenever we do have a lot of different types of bacteria. And we need to grow just one of them. An example of selective media is McConkey agar. This culture media will allow to grow just gram-negative bacteria. And as well as this culture media is used for diagnosis of intestinal infections, and as well as we can know that the majority of intestinal infections are caused by gram-negative bacteria. Meanwhile, the majority of normal flora in the GTI, in the GIT, gastrointestinal tract, are gram-positive. Whenever we'll take some samples from GIT to cultivate bacteria and to find out the pathogen which produced one disease or another one, we will use selective media because the intestinal tract contains a big amount of different bacteria. So in this case we'll need to select just gram-negative bacteria. In this way we will be able to find much faster the disease caused by this bacteria. Meanwhile, differential media will help us to make difference between different species of the same genus and how it can help us. Here is an example of differential media is triple sugar agar. And differential media will allow to grow different species of the same bacteria on its surface. But if you will look under the microscope on these bacteria, you will not see any difference between different species of these bacteria. Meanwhile, if you will grow them on differential media, since they have the same appearance but they have different metabolism, all of them will use different sources of energy, different sources of carbon. Some of them will use glucose, some of them will use lactose, and so on. Some of them will produce H2S, some of them will not produce it. And since they will use or not use some kinds of sugars, it will change the pH of the culture media. Since it will change the pH of culture media, it will change also its color from pink to yellow. But if it will not consume some kinds of sugars, it will not change the color of the media at all. Meanwhile, if they will produce H2S, the color of culture media will become black, from green to black. And also, it can show does they produce any gas or not. So you will see the space in the bottom of the tube, you will see the space with gas, the empty space, if it produces any gas. And you will not see this empty space if it will not produce any gas. So again, if you will use the microscopy for different species of bacteria of the same genus, you could not see any difference among them. Meanwhile, if you will use culture media, differential culture media, and you will cultivate these bacteria on this culture media, you will see the result of different metabolic processes which can happen in different bacteria. So these metabolic processes will change the color and appearance of this culture media, and in this way you will see the difference and you will be able to make the right diagnosis. You will be able to appreciate which kind of bacteria, which species started this disease. Also, we have transport media. Transport culture media is some kind of media which actually has a purpose to preserve the bacteria when we need to transfer it in the lab. So as well as you know, not every kind of bacteria is resistant enough outside of the host. Not all of bacteria will survive on their way from the source till the lab. And not every time we have lab nearby the source. So sometimes we need to take the sample from the infected person and to bring it in the lab. And this way could have different this way could have different length. Sometimes the laboratory is nearby, sometimes the way could take few hours. And these few hours could be enough for some bacteria to die until they will be brought in the lab. So for such cases, if we suspect an infection with pretty high sensitivity bacteria, for such cases we apply transport media. We will put the bacteria in this transport media, which will allow bacteria to survive until we'll bring it in the lab. And whenever we are talking about anaerobic media, it is that kind of media which provides anaerobic condition for anaerobic bacteria. So in first cases, we have media which has access to oxygen, unless we'll put it in anaerostat, the device which takes out all the oxygen. But also, if we don't have this device which provides anaerobic condition for anaerobic bacteria to cultivate them, we have different kinds of anaerobic culture media. And usually we are using for such purposes oils. So basically it will be a culture media in the tube which will be covered with a layer of oil. So through this oil, the oxygen will not be able to penetrate inside the media. In this way, the anaerobic bacteria will be able to survive. Here you can see Levenstein-Jensen media. It's a solid culture media which is used for Mycobacterium tuberculosis. Well, whenever we are talking about selective, media used for Mycobacterium tuberculosis, we can say it is a selective culture media because it will inhibit growth of both gram-negative and gram-positive bacteria. And it will allow to grow just Mycobacteria. Meanwhile, Mycobacteria is specific kind of gram-positive bacteria which is covered by specific mycoic acid. And this mycoic acid is a kind of waxy membrane which makes these bacteria to be resistant to acids. Why this culture media contains penicillin or others? So penicillin will inhibit growth of other bacteria, but at the same time, in this culture media can be tested on sensitivity of these bacteria for different drugs. So usually we'll take a lot of tubes like this, and we will cultivate on all of them the bacteria. So in the beginning we'll take the simple one, and we will put here some samples. But on other tubes we'll have dissolved more antibiotics except penicillin, and others. We'll put there some different antibiotics which are used in the treatment of tuberculosis. So in such case we will have few tubes with the same sample, and we'll put it to grow, to multiply in thermostat. So it will be the test for sensitivity of Mycobacterium for different antibiotics. In order to find out if this kind of bacteria is multi-drug resistant or not. And here you can see some examples of culture media. Well, hopefully this basic first information will be enough for you to get ready for our next practical lesson. So, wish you good luck and see you on the next lesson. Bye bye.